Hey guys, welcome to your first lecture video, a tour of the cell. So today we're going to talk about a couple of different aspects of cell biology. We'll talk about how people study cells, and we'll talk about the different types of cells that exist. So I always like to start with a little bit of a historical perspective when we're talking about science. So you may think of cell biology as something like microscopy. So this should be sort of in the category of cytology. But many, many years ago, scientists studied cells in a lot of different ways, microscopy included. Biochemistry was a very popular thing to do, isolating enzymes and proteins and characterizing their compositions and their activities. And on the other hand, we have genetics, which is going to be sort of the Gregor Mendel line of thought. Now, in 2020, we know that all of these things work together. We know that you can see cells in a microscope. They have structure. We know that they have enzymes and organelles that are going to be performing certain types of activities, and we also know that they contain genetic material. So though everyone sort of studied these separately, we can eventually see how the paths intertwine to give us what we now know to be modern cell biology. So let's just hit a couple of the high points. So of course, you always have to start microscopy by reminding everyone about Robert Hooke observing quark cells. Okay, so that was kind of the very beginning of the cell biology that we think of as microscopy. Not too long after that, we get improved lenses, and Lewin Hooke allows us to see things a little bit more clearly. We then, after studying cells for quite a while, so you can see that we've made quite a jump in time here, that we start to formulate cell theory. So what is cell theory? Let's just refresh our memory. So expanded cell theory means that all organisms are made of cells. The cell is the simplest collection of matter that can live on its own. Cell structure will be correlated to its function. We'll be talking about this a lot more later. And all cells are related by their descent from earlier cells, and that's where the genetics component comes in. So let's head back to our timeline and take a look at what's happening. Okay, so we're still a long time ago, more than 200 years ago, looking at cells for kind of almost the first time. And along the biochemistry route, we start to get some other information that's going to help us understand. So we start to get by linking organisms to specific processes. This is where we ultimately get pasteurization. We start to learn more about fermentation, again, other biochemical processes that are taking place as part of cell biology. And then we start to learn more about the glycolytic pathway and the TCA cycle. Okay, so we'll save the details of that for chapter 9. Along the way, we start to get the genetics information filled in. This is Gregor Mendel first formulating the principles of genetics. The discovery of DNA as a molecule actually comes after that. Just for reference, around 1952 is when we get the shenanigans of Watson and Crick. So though we did discover that DNA is a molecule, we didn't know really what exactly it was doing or its shape until much later. We can start to see that we get the invention of microscopes with a lot more power for us to see. We start to develop technology that will allow us to study proteins in living cells. So we'll be talking about fluorescence microscopy today as well. And we start to get light microscopes that begin to surpass theoretical limits. So the take home from this is really to think about sort of the perspective people had historically and where we're going, our future trajectory with modern cell biology. So no longer is cell biology just simply looking in a microscope, but it's going to have a lot of elements of biochemistry and genetics as well, because these are all going to be related to the function of cells. But we will go ahead and start our lesson on how we study cells with microscopy, because it is one of the quintessential sort of tools. Okay, so you know that cell biology, we're using microscopes. It's important to have at least an idea of how these guys work, so we'll just do a brief run through. You can see here's our microscope. We'll be dealing with these some in lab, and the basic way that these work is you can produce a very thin sample on a slide. So here's going to be our slide up here, and some type of light source. So you get one of the fancy microscopes, it might have a light bulb. If not, you might actually get a mirror that you use to reflect light up into the sample. We're going to pass light through the sample. Okay, so it has to be thin enough to pass through. Ricochet around, objective lenses, eyepiece lenses, get the desired magnification, and then we can see the image in our eyes. So this is going to be the way that basic light microscopy is going to work. And we're going to talk about several different types of light microscopy and talk about the strengths and limitations of each. Okay, so when we consider strengths and limitations of microscopy just in general, we sort of think about three parameters. This is going to be magnification. So how many times can an image actually be enlarged? I think this is probably one of the most straightforward aspects of microscopy. Of course, we want to make the sample larger. Contrast is another issue. So how different are various parts of the image? Okay, we'll be looking at some examples of this on the next slide. And resolution, we know a lot about resolution. If you look at pictures that were taken in the 90s, you know that they are a lot more fuzzy than pictures that are taken today. So that's an increase in resolution. When we're magnifying things, we have to get a good balance between making it large and being able to still clearly identify things in the image. So each type of microscopy, again, will have its own strengths and weaknesses. Some of them will give us enough magnification, but not enough contrast. Some of the times we can look and see, okay, magnification is okay, but resolution is not good. So that's what we will be looking at. So let's actually start with contrast. And here we'll see several different types of light microscopy. So on this first slide, the type of light microscopy we're going to be looking at is called Brightfield microscopy. This is the one that includes the types of microscopes that we use in the teaching laboratories today. So just to kind of plug them in, turn on a light, look at a specimen. Okay. So if you guys have looked in microscopes before, you know, sometimes it's a little bit difficult to see. So this is a typical Brightfield type of image. And these are probably some type of epithelial cell, skin cell type of a thing. And the light has to pass directly through the specimen. But you can see, even though the border's okay, there's not a lot of contrast in what's going on here. So a lot of times what we can do to improve that with Brightfield microscopy is actually stain samples. Okay, so here you can see a lot more detail. You can't see a lot of organelles, but you can find the nucleus. You can see where the actual shape and structure of the cell is. So this is a lot better for viewing. One of the limitations of this type of microscopy, however, is that we have to fix the sample and stain them. This generally means that the sample is not alive, so we can't view any dynamic processes within the cell. In order to try and get around this issue, we can also play some tricks with light. So instead of using a chemical stain, we can use sort of optic techniques with phase contrast and differential interference contrast microscopy that will allow us to have a little bit more contrast, but still have these unfixed living cells. So you can see that if we just want to look at what's happening along the whole cell, then no big deal. If we want to get a lot more detail, we may have to choose another type of microscopy. Okay, so you can see that there are a lot of different types of light microscopy so far and Brightfield microscopy, but if we want a lot more detail, we'll have to go in a little further. So the next type of microscopy that we'll talk about is fluorescent microscopy. So you can see very clearly, it's a very vivid image, and you can see we've got a nice nucleus stained here. The green stain likely to be something like microtubules. I would bet that this red stain is probably actin, which we'll be talking about at the end of chapter 6. So all of these structural elements within the cell. But you can see that there's a lot more clarity going on. So fluorescent microscopy works by tagging molecules within the cell with fluorescent dyes. Sometimes these cells can be engineered so that they're still alive while we view them, but some of the time we actually have to use fixed specimens as well. So they can be used for living cells, but not always. Okay, so there is a little bit of flexibility with that. Confocal microscopy is similar in a lot of ways to fluorescent microscopy. Okay, so with fluorescent microscopy, we're still using light. This is light microscopy. We're likely going to be shining one wavelength of light onto the sample. Confocal microscopy is just going to be a variation of that. So on the next slide, we'll see the differences. Okay. So at the top, this is an example of confocal microscopy. At the bottom, a standard fluorescent image. So hopefully you're able to tell that even though the magnification and the contrast are largely the same, the clarity resolution in this confocal sample is a lot better. Okay, so how do we achieve this? Is there something different that we're doing? Not really in terms of sample preparation. It turns out that this has more to do with the microscope that we're using and how it's going to be taking pictures of this image. So to understand what's happening, you have to think about the fact that even though these samples are very flat, they still are 3D. So as we focus the microscope, we may see just the top of this image. We may see something that's kind of halfway through the middle, or we may see sort of the bottom of this image, if you think about this as a 3D structure. Okay, and when we think about this having a little bit of depth, you can maybe compare it to a photograph. So you can see picture on the left. There's a lot of stuff going on here. There are some things in the background. There's some things in the foreground. There's some things kind of in the middle. Okay, so if we want, we can focus our camera only on the middle part. Okay, so that would be like the middle of the sample. There's still stuff to be looked at in the back and the front, but our vision is focused here. That makes things blurry in a different focal plane. With confocal microscopy, what we're doing is actually focusing in layers. So we may take a picture closer to the front, a little bit further back, a little bit further back, a little bit further back, through the middle, and eventually, even though I don't have a clear picture of these little trees, you would get to the point where your focal plane was on the actual trees. So focusing each time through a different part of focal plane. Once this is complete, a computer will actually put these images back together so that it looks clear. So this is actually just the result of taking lots and lots and lots of pictures progressively down through the sample. So this is a really good way to get a clear image. Okay. So we've talked about types of light microscopy. We've talked about contrast. Magnification is always the goal. We've talked about resolution. And with all of these types of microscopy, particularly with the fluorescence images that you've seen, we're able to really get a lot of intracellular detail. But you guys know that there's a lot more detail and smaller structures that we might want to be able to see. Okay, so we then have to think about the relative sizes of things. Okay, so here is sort of a scale from things we can see with the naked eye, light microscopy, and what happens when we get even smaller than that. Okay, so I'm not typically a fan of having you guys memorize these numbers, but more just kind of think about your experiences. You know that you could see a person without any help from a microscope. You know that you could see a chicken egg without any help, and a frog egg probably too. Okay, we know that we've had enough experience with light microscopes that we can see cells. We just saw them on the previous slide. Okay, we might be able to see some organelles, some intracellular structures. Okay, so we know that a light microscope can help us see those. When we get smaller than what a light microscope can help us see, we need to employ the help of an electron microscope. Okay, so the difference between electron microscopes and light microscope is that the beam shining through the sample in light microscopy is exactly that. It's composed of light, either just full spectrum white light from a light bulb, or maybe one wavelength of light in fluorescence microscopy. In electron microscopy, we will literally be shooting a beam of electrons at the sample. So you can't see electrons. They're not the same thing as light. And the reason this works is because this electron beam has a shorter wavelength, and that gives us better resolution so that we can magnify things and still get that clarity that we need. So we can really see things like viruses, even ribosomes, larger pieces of DNA, some larger proteins. We can see a lot of stuff with the electron microscope, okay? So let's just kind of, again, think about what are the kinds of things that we can see, okay? And let's start to, in our minds, get a relative idea of sizes. So I think it's always important for us to understand the relative size of things. So again, we don't feel the need to memorize these numbers. We just know which things are smaller than others just by default. So we'll start as small as we can go, okay? Things like oxygen and carbon dioxide. You can see they're hanging real far down here. We may not even be able to see them, all right, at 360 and 230 picometers respectively. Okay, so we're down here almost near atom territory. Just above that, here is SARS-CoV-19. So this is COVID-19. This is about 120 nanometers in diameter. Okay, so we're like somewhere right in here. Okay. Red blood cells, for reference, these were about 6 to 7 micrometers, so you can see several orders of magnitude larger. Okay, hanging up here, you can see these fairly easily with a light microscope. Respiratory droplets. So there's been a lot of talk about respiratory droplets in the context of spreading COVID-19. So a lot of research has shown that these respiratory droplets can be anywhere from about 10 to 100 micrometers, okay, just on average. So some of them are really small, some of them are really large. Just to compare, the pores in one layer of cotton fabric, depending on the weave, the type of cotton, can be somewhere between 20 and 100 micrometers. So here are 3 types of cotton. You can see the pores between them. Okay, the scale bar here is 300 micrometers, so that's where we're getting this estimate. Okay, so you can see the sort of scale. All right, I like to compare this again to the size of respiratory droplets, but also to the size of oxygen and carbon dioxide. Okay, again, we can keep moving up the scale. We know chicken egg, I have seen one of those before without a microscope. So again, you don't have to memorize the sizes. It's important, again, to get a good visual in your mind about what the relative sizes are. Atoms, molecules, more complex molecules, larger protein structures, viruses, bacteria, all the way up through sort of the cellular area back up into naked eye territory. Okay, so we've talked about relative sizes of things, and now we have a really good idea of what types of things we may need an electron microscope for. So let's talk about a couple of different types of electron microscopy. So first, at the top, have what's called scanning electron microscopy. This is kind of what you think that it would be. We sort of scan along the top of these samples, okay? This gives us a lot of surface detail, okay? And the way that we're able to see this is actually by staining the surface of the cell with metal particles, and the electron beam will hit it, and we'll get a signal back to a computer, and the computer will generate the image. So you can't actually see this. This is created by a computer. So scanning, I like to think of for a surface. You can really see the cilia in this picture. This is beautiful. The other type of electron microscopy that we're dealing with is transmission electron microscopy. So you can see we've got a lot of detail here, and these are usually involved with cross sections. So this is kind of the same type of cell as above, but we've sort of sliced through it. So we're looking at a cross section instead of the surface. Okay, so you can see all these cilia, what's going on inside. Again, at the end of the chapter, we'll be talking about how cilia are going to actually be structured. You're going to see some of the cytoskeletal elements that compose this. But for now, this is really nice. Okay. These give you a lot of detail, but because of the way they have to be prepared with the heavy metal stains, and gold particle stains, shooting them with electron beams, this will damage and kill our cells. Okay, especially this guy's a cross section. So we really can't use this for dynamic processes. So sometimes we have to sort of sacrifice. Do we really need to see this dynamically? Does the cell need to be alive? With how deep do I need to get in there and really see the structure? Okay, so one of the things that we'll be focusing on, you guys will need to know several aspects of this microscopy, but more importantly, understanding how to choose the correct tool for the job. Okay, so you can have, all kinds of tools. You can have a hammer. Is a hammer the best tool for the job? That depends on if you're trying to nail something in or if you're trying to cut something or whatever job you're doing. The same thing with microscopy. Each one performs a certain job well, and it has its own strengths and limitations. So what we're going to be learning to do is actually understanding why each type of microscopy should be used. If we don't really care about contrast and we want to see living cells, we don't need to get in there that deep, we might choose some type of light microscopy. If we want to see cells in a little bit more detail and at least have the potential for them to be alive, or if we're looking for a specific cellular structure to be really clear, like all of those red and green elements we saw stain, we might choose fluorescence. If we need to really see detail that's so small, either on the surface or as a cross section, we're going to need to transition then to electron microscopy. So that's what I'm going to want you guys to be thinking about. Okay, and of course, other cool things you can do with microscopes. This is just really a field that's way cooler, I think, than people think it is. But if you're going to make microscopy even better, the obvious thing to do is combine it with lasers. So a couple of things you can do by combining lasers with our actual microscopy is you can use the laser to move things around. So just use them like, sort of like optical tweezers to look on the slide and actually manipulate and move things around. That idea can also be used to dissect very small areas. So you can see here, you can select a region we're interested in, go ahead and cut it out right along the lines, take it, put it in a tube, whatever else we want to do with it. This can also be used, so this is a plant development timeline. We can also use this to dissect out very small parts so that we can analyze things like gene expression all throughout development. So this is a very cool thing that we can use microscopies in conjunction with lasers. So we can head back thinking about the way that we study cells. So we've talked a lot about microscopy. We've learned about Brightfield microscopy with or without stain. We've learned about our different types of optical tricks that we can use, like phase contrast and differential interference contrast. We've learned about fluorescence / confocal microscopy, all of which use light in one way or another. We've also learned about scanning and transmission electron microscopy. So just to add kind of a biochemical element to this, we'll be talking about some of the genetics elements a little bit later. And to tie this into our second week of lab, again, we can also use biochemistry techniques to study cells. So this particular slide, just to remind you that we can actually isolate out organelles by using differential centrifugation to get increasingly small pieces of the cell components, nuclei, mitochondria, chloroplasts, pieces of lipid membranes, ribosomes, etc. Okay, we can get a lot of really small things here. Okay, so if you want to know more about differential centrifugation, I posted a video that goes with week 2's lab materials because we'll be performing that in lab. Okay, so more detail on that, but just to connect this up with what we'll be doing. In the next video, we'll be looking at what types of cells exist.